Hello?
Hi, Anthony. This is Tony. Is this the right number and right time to call?
It's fine, Tony.
Okay. So what's going on? How are you?
I'm doing very well, and yourself?
I'm fine, Anthony. So any update from the last time we spoke about approval of glyphosate by EPA and problems that you found?
I have one update. I received a laboratory report back from the technician today on analysis of glyphosate in laboratory feeds in rat chow and rodent chow that's fed to the animals during scientific studies.
Okay.
And the results were pretty significant. All of the rat chows, all of the laboratory feeds that I analyzed are contaminated with glyphosate.
Oh, really?
I talked to Purina and asked them if they could send me some certified chows, and they sent me some samples, and I put them in the lab, and the technician analyzed them for glyphosate. I suspected that the rodent diets and all the lab diets being fed animals since they started using genetically engineered feed and spraying crops with glyphosate, I suspected that these diets would be contaminated with glyphosate herbicide. And of course, as you know, it only takes low levels of glyphosate to cause profound changes in the biology of animals and us. So what I found in these laboratory diets is going to significantly change how we view scientific experimentation with animals, because the results that have been recorded since they've been using contaminated chows may not actually be the true picture of what's taking place. Glyphosate enhances and amplifies the effects of other chemicals. So all of the studies that have been done, people are going to have to start taking another look at their materials. If they saved chow from their lab experiments, they should most likely analyze that for glyphosate and its metabolite AMPA. I found significant, like I said, quantities of glyphosate and the metabolite AMPA in all three of the rat chows that I analyzed. One of the rodent chows was a certified rodent diet number 5002, and that's a very popular lab chow for feeding rodents. And the ingredients in that chow are ground corn, dehulled soybean meal, whole wheat, wheat middlings, dried beet pulp, wheat germ, cane molasses from sugar cane, ground oats, soybean oil. All of these that I just mentioned, those are all sprayed with glyphosate when they're produced on the farm. And all of those contain glyphosate and AMPA as a contaminant. So they formulate these together to make this particular rodent diet. And when I analyzed that chow, I found 0.65 parts per million of Roundup and 0.35 parts per million of the metabolite AMPA in that particular chow. Then I took a look at another chow. I took a look at the Purina 5LG3, and that's an advanced protocol certified rodent diet. And it's recommended for growth and reproduction of rats, mice, hamsters. It's a pretty popular formulation. It too consists of ground corn, ground wheat, wheat middlings, corn gluten, dehulled soybean meal, dried beet pulp, soybean oil. These things, again, as I mentioned in the 5002 diet, these are all contaminated with glyphosate. They're all farmed, they're all sprayed with glyphosate, they all carry the herbicide and its metabolite. Then I looked at a third popular rodent chow, and that's the certified Pico Lab Rodent 20. It's also from Purina. It's the 5K75 diet. Has 20% protein formulated for feeding rats, hamsters, and mouse colonies. And again, this particular diet starts off with corn, dehulled soybean, wheat middlings, ground wheat, cane molasses, wheat germ, dried sugar beet pulp, and all the vitamins and things that they add to these formulations. But again, another diet, it came back positive, that 5K75 diet, looking at the lab printout here for the 5K, we got 0.57 parts per million, glyphosate 0.27 parts per million of the metabolite AMPA. And that other diet, which was the 5LG3, we got 0.37 milligrams of glyphosate per kilogram, which is 0.37 parts per million. And we got 0.10 part per million of AMPA. So again, all three lab diets totally contaminated. This is going to be a problem for the scientific community that they need to address, because as I mentioned in my third paper on glyphosate, I cited a bunch of patents that show that glyphosate is a synergist. There was an interesting study that was done. I was reflecting on it tonight. I pulled it up, and it's it's about folic acid supplementation. And there was a study done around 2010, and they showed that it promoted mammary tumors in rats. And they mixed the folic acid with the Lab-chow. Now, folic acid has been it's been a contentious issue. Some people say that it doesn't cause cancers, and other people say that it does cause cancer. And this recent study, I think, as I mentioned, it was around 2010. They showed that folic acid supplementation promoted mammary tumors. Now, when you look at that study, you know that the Lab-chow that they fed the animals was most likely contaminated. Glyphosate contamination of Lab-chows began when they started using genetically engineered corn and desiccated wheat, when they started using sugar beet pulp, which is genetically engineered and sprayed with glyphosate. When they started putting all those things in the Lab-chows, that's when all the Lab-chows became contaminated, and that was well before this folic acid supplementation promoting mammary tumor study was done. So these people are probably going to have to revisit their experiments. They're probably going to have to redo them with clean feed that doesn't have glyphosate in it, because folic acid may or may not induce mammary tumors. We don't know from that study, because we know that all the chows from the distributors have been contaminated over the years. Now, if we go back to the early history of when Monsanto did the original glyphosate tests in mice, rats, rabbits, and dogs back in the late 70s to early 80s, even the 1990 long-term rat study, those lab diets, they were all formulated the same. They were all provided by Purina. That's who provided the lab diets for all the Monsanto work. But those diets wouldn't have had glyphosate in them. So any of the findings that were in there are probably accurate. The tumorigenic growth that they found, the cancers that they found, are most likely due to the glyphosate.
That is because glyphosate had not yet been used in the growing those crops, which are part of the diet, right?
That's correct. So the treated or dosed animals would have been the only ones seeing the glyphosate. We wouldn't have had to worry about the control. In the control feed, what we would have had to worry about would have been the organophosphate pesticides, which are cholinesterase inhibitors. Residues of malathion or lanate, carbaryl, things like that. Those things could possibly have contaminated the controls and caused the controls to indicate findings. However, when as I've been looking at those Monsanto studies, I find that most often, if the control was positive for a particular gland or organ tumorigenic growth, most often the glyphosate-treated animals had greater incidence than the control, which potentially could show that glyphosate had a synergistic effect with whatever was in the chow. So, but the story here is that science is going to have to take a look at the feeds that they're feeding animals in animal studies. The chows are contaminated. 90% of the corn, soy, canola that are being produced today, genetically engineered, they're all sprayed with glyphosate. They all carry contamination. So to feed those to the laboratory animals and expect good results, you can't expect a good result with a contaminant. And they don't test for glyphosate in the lab chows. My discussions with Purina in St. Louis, Missouri, I asked them if they tested for glyphosate or glufosinate, and they said they don't. They do batch analysis, and it's not every bag that's analyzed. It's there are lots that they'll do a spot analysis here and there, and they'll look for some of the older pesticides like malathion or some of the fungicides, but they definitely don't analyze for glyphosate.
And they were not asked to analyze for it either.
They're not asked to analyze for glyphosate either, but they should be. The EPA should be taking a good hard look at this because the studies that they're accepting are all questionable now. Even the Food and Drug Administration, they need to be very, very concerned. The National Institute of Health needs to be very concerned. All of these laboratory animal-based studies, they need to be concerned with glyphosate as a contaminant because glyphosate induces all kinds of diseases. It disrupts the animal's bacterial balance, and it causes overgrowth of pathogens. It disrupts the CYP450 enzymes as well as other enzymes, which are our first-line defense against metabolizing xenobiotics out of our biology. When we destroy our beneficial bacteria species, we disrupt the manufacture of vitamins, like all of our B vitamins, vitamin K, even some vitamin C, fatty acids, essential amino acids like tryptophan, phenylalanine, and tyrosine, even methionine. Those are all disrupted by glyphosate. So feed could be making the animals sick. Not could, would be making the animals sick. And again, enhancing the effects of other chemicals that would be added to with it in the feed.
Fantastic. So are you going to write a paper on it, or just publicize it, or what?
I'm just giving you an update. This information will be included in our next paper about glyphosate and cancer. That paper will be coming out early this fall. We're working on it now, and this will be part of the subject that we'll be discussing as to proper laboratory protocol. The laboratory protocol currently in use needs to be overhauled, and that includes analyzing the feed for these herbicides, as well as stopping the use of historical controls, adding those into the experiment to negate the results. That's not good laboratory practice. That's the same as polluting a study by contaminating a control. Same difference. The practice of using historical controls needs to be banned from science, along with contaminated feed that's contaminated with glyphosate or other herbicides. We analyze for it to verify our assumptions, and then we pass the information on. And so the scientific community should take notice of this, and they should start looking into it before they conduct their experiments.
Fantastic.